Hello everyone. So today we're going to dive straight into the glucose metabolism. Particularly we're focusing on glucose homeostasis. So we're going to be focusing on how the body regulates blood glucose levels, how it prevents hypoglycaemia or hyperglycemia through hormonal control, specifically glucagon and insulin. We're going to look at the signal transduction pathways that follow glucagon and insulin. We're going to focus on the kind of biochemical mechanisms behind glycogenesis and glycogenolysis, and how glycogenesis and glycogenolysis can be used to kind of regulate blood sugar levels. We're also going to be looking at later on in the video, we're going to look at how kind of neuronal control can also kind of govern pancreatic insulin or glucagon release. We're going to look at the role of the hypothalamus in glucose homeostasis, and we're also going to look at the effects insulin and glucagon has on several metabolic processes like lipogenesis, beta oxidation, gluconeogenesis, glycolysis. This part one is only focusing primarily on glycogenesis and glycogenolysis, as well as insulin and glucagon's role in those processes, as well as adrenaline signaling in muscle cells and glycogenolysis and glycogenesis in muscle cells. The next video is going to focus more on glycolysis and gluconeogenesis. So before we kind of move on to the rest of the video, I just want to quickly touch on a few metabolic pathways just so people are familiar with them. It will make more sense as we're going on in the video, but I think it's important to kind of be aware of these pathways. And of course, I'm going to go over them in a lot more detail in other videos, looking at the enzymes, their metabolic intermediates, structure of all these intermediates. But for now, I think it's important to just kind of be aware of them. So the first one you'll hear a lot is gluconeogenesis, and that is a process in which cells will use the carbon skeleton of certain glucogenic amino acids to generate or build glucose. Okay, so they convert amino acids, glucogenic amino acids like alanine, into Krebs cycle intermediates like oxalate acetate. And then that oxalate acetate can be converted into phosphonyl pyruvate, and then you get your glycolytic intermediates, and you can build your 6-carbon sugar, your glucose. And that's typically done in a fasted state, or when you're having high protein diet, like a high protein low fat diet. The next pathway is fatty acid synthesis or lipogenesis, which is the creation of fatty acids from pyruvate, or from glucose, essentially indirectly from glucose. Now, of course, pyruvate can be converted into acetyl-CoA. Then acetyl-CoA carboxylase can convert acetyl-CoA into malonyl-CoA, and malonyl-CoA is that rate-limiting step in initiating fatty acid synthesis. And this is really important because glycogen storage can only take our body so far. We can't store too much glycogen in the liver. So we can't store too much glycogen, and the glycogen in muscles is used for energy. So a lot of that glucose can indirectly be converted into fat if we have an excess of it. Okay, so that's lipogenesis. Now the next pathway that I'm going to just touch on briefly is beta oxidation or lipogenolysis, and that's the breakdown of long chain fatty acids into ketone bodies, okay, like hydroxybutyrate. And again, this is usually done in a fasted state or when you're in a high protein state, and that usually beta oxidation provides the energy for gluconeogenesis when dietary glucose is absent. So beta oxidation is the breakdown of those fatty acids, those long fatty acid chains, into your ketone bodies like hydroxybutyrate. Okay, and then that hydroxybutyrate can be used for energy. Now, the next pathway I'm going to talk about just briefly is glycolysis, and that we'll go over that in a lot more detail in part 2, or in part 3, sorry. And glycolysis is the breakdown of your glucose 6-phosphate into pyruvate, okay, and you get a net gain of 2 ATP molecules. And of course, that acetyl-CoA can enter the Krebs cycle, and then you get your generation of your reduced coenzymes NADH and FADH, and they can donate electrons to the electron transport chain, oxidative phosphorylation, and they can produce the vast amount of ATP that is produced during cellular respiration. But they're the kind of metabolic pathways that I think is important to understand when looking at the bigger picture, and you'll see why as we kind of move through these slides. So before we kind of dive into the glycogen metabolism, glycogenolysis and glycogenesis, I think it's really important to understand the concept of homeostasis. And homeostasis essentially specifically blood glucose homeostasis. Homeostasis refers to mechanisms in which the body can maintain a stable condition. Okay, and in the context of blood glucose levels have to remain at a consistent concentration in the blood. If they get too high, then it will induce something called hyperglycaemia. And if it gets too low, consequentially, you can get hypoglycemia, and both of them can be detrimental to your health. So your body has to find a way to be able to regulate blood sugar levels. So for example, once you've eaten a sugary meal, your blood sugar levels will raise, especially if you've had a carbohydrate heavy meal. Okay, and conversely, if you're in a fasted state, your blood sugars are going to be low, and you're going to risk hypoglycemia. Okay, so your certain organs in your body, like your pancreas, will help maintain glucose homeostasis along with your liver. Okay, and then you have structures like your hypothalamus. Okay, neural circuitry also plays a role in kind of sensing kind of physiological or physiologic glucose concentrations based on ATP levels, based on levels of glucose in the blood. Okay, and those cells in the hypothalamus can then signal through the autonomic nervous system, parasympathetic or sympathetic, and stimulate the release of either glucagon or insulin. Now, insulin will lower blood sugar levels and promote glucose storage, facilitate glycogenesis, whereas glucagon will do the opposite. Okay, it will facilitate hepatic glucose output, glycogenolysis. Okay, raising blood sugar levels in a fasted state. So just so we kind of like understand that concept, the concept of homeostasis, to kind of move forward and apply this to glycogenesis and glycogenolysis. So the first hormone we're going to be talking about is insulin. Now, a lot of people would have heard of insulin. It's a very famous hormone and it's world renowned. You know, even people who don't really know much about biology would have probably heard of insulin. So insulin is a peptide hormone, so it's made up of many chains of amino acids. Okay, and it's produced, okay, it's not made from cholesterol like steroid hormones. It's produced through gene transcription and translation. Okay, so it is a peptide hormone, and it's secreted from beta pancreatic cells. Okay, in what's called, you know, we have these clusters of cells in what's called the islets of Langerhans in the pancreas that contain alpha, beta, and delta pancreatic cells. Now, alpha pancreatic cells secrete glucagon, and beta pancreatic cells secrete insulin. Okay, and when blood sugar levels rise, insulin is secreted from these beta pancreatic cells. Okay, and insulin will then bind to receptor tyrosine kinase receptors on, say, hepatocytes, on your liver cells. Okay, it will initiate a signal transduction pathway. It will result in the activation of specific enzymes, and then it will facilitate or promote glucose uptake and glucose storage. Okay, and then, as we know, glucose is stored in the liver, in your fat cells, your adipocytes, and in your muscle cells, your myocytes, via glycogenesis. Okay, and that kind of facilitates glucose storage. Now, these beta pancreatic cells respond directly to blood glucose levels, and I'll talk about that in the next slide, but they can also receive signals from your autonomic nervous system, so your sympathetic and parasympathetic nervous system, which has efferent nerves that go from the kind of hypothalamus, and they project through the spinal cord, and then from the spinal cord into the periphery to your pancreatic tissue. So your nerves can directly kind of signal your pancreatic tissue as well. So your hypothalamus plays a really important role and kind of potentiates and amplifies insulin secretion or glucagon secretion, depending on blood sugar levels, depending on whether you're in a fed or in a fasted state. So, as I said before, the beta pancreatic cells are responsible for the synthesis and secretion of insulin, okay? And the alpha pancreatic cells are responsible for the synthesis and secretion of glucagon. And those delta cells will regulate the beta and especially the alpha cells through paracrine inhibition. Now, the pancreatic tissue has what's called the islets of Langerhans, and that is composed of the beta, alpha, and delta cells. And these cells can directly respond to blood glucose levels, but they also receive nerve input as well. They have nerve circuitry from the autonomic nervous system, and this is kind of governed by the hypothalamus, particularly the arcuate nucleus to be precise. And those neurons have kind of glucose sensing capabilities. They can sense ATP levels. They respond to ATP levels because, of course, glucose levels are directly correspond or correlate to ATP levels because of cellular respiration, the relationship with glycolysis, okay, and cellular respiration. But these cells can also directly respond to blood glucose levels as well. So they're regulated via our nervous system, but they also respond to blood glucose levels, whether you're in a fed or a fasted state. So either way, that nerve circuitry is going to potentiate or amplify glucagon or insulin release, depending on depending on blood sugar levels. Okay. But kind of moving back, taking a few steps back to the beta pancreatic cells. So in the islets of Langerhans, one of those cells are the beta pancreatic cells. As I said, they synthesize and secrete insulin in response to raised blood glucose levels or hyperglycaemia. So if you've just had a high carbohydrate meal like pasta, if you've had mashed potato, if you've had any kind of carbohydrates, where in the digestive system those carbohydrates are broken down into sugars, into glucose, it's taken into the bloodstream. Okay, and these beta pancreatic cells, like most cells, have these protein transporters called GLUT transporters. Okay, and they're GLUT2 in beta pancreatic cells. Of course, you also have GLUT4 transporters as well, but GLUT2 in beta pancreatic cells. Now, these GLUT transporters allow for the passive diffusion of glucose into the cells from the vascular system, from your blood, okay, into these cells. So in the lipid membranes of these beta pancreatic cells, they express these GLUT2 transporters, and that allows for glucose into the cell. So once blood glucose levels are higher after you're in a fed state, more glucose is going to go into these beta pancreatic cells via these GLUT2 transporters. And of course, when glucose goes into the cell, most of the time glucose is used for cellular respiration. Okay, glucose is broken down into pyruvate in a process called glycolysis. Okay, and that has a net gain of 2 ATP molecules. And of course, if you want to take it a step further, pyruvate, acetyl-CoA can go into the Krebs cycle, okay, generate a lot of coenzyme FAD and NAD, and those NAD and FAD act as electron donors in the mitochondria to the electron transport chain. You get oxidative phosphorylation, and you get a lot of ATP being produced in oxidative phosphorylation. So glucose comes into these beta pancreatic cells, and naturally ATP levels will increase. Now, another type of protein channel these beta pancreatic cells have in their lipid membranes is ATP sensitive potassium voltage gated channels, and that usually allows for the flow of potassium out from the intracellular space into the extracellular space. Okay, and you have a polarized membrane. There's more potassium outside the cell than there is inside the cell, and that's because these channels are open. But these channels are sensitive to ATP. They're ATP sensitive. So when ATP levels rise, and when adenosine triphosphate levels become greater than adenosine diphosphate levels, so the ATP ratio is greater than the ADP ratio, the ATP to ADP ratio, okay, these voltage gated potassium channels will close, and that prevents potassium from leaving the cell. So you get what's called depolarization. As I said before, the membrane is polarized because there's more potassium in the extracellular space. But when these voltage-gated potassium channels close, okay, you get more potassium accumulating in the intracellular space. So you get what's called depolarization. Now, another type of channel that these beta pancreatic cells have in their lipid membrane is also the voltage-gated calcium channels. Okay, and typically those voltage-gated calcium channels are closed when the membrane is polarized. But when you get depolarization, you get those voltage-gated calcium channels open, and they allow for the influx of calcium into the cell, an intracellular influx of calcium. Now, a lot of people are kind of familiar. Calcium acts as a really kind of potent chaperone, if you like, and it allows for vesicular or granular migration. Now, insulin, once synthesized and secreted through protein synthesis, is stored in these granules, kind of the same way that neurotransmitters in neurons are stored in vesicles. And again, depolarization in neurons results in the opening of those voltage-gated calcium channels at the synaptic cleft, and it stimulates vesicular migration and neurotransmitter exocytosis. And it's the same principle when calcium goes into these beta pancreatic cells. Okay, when you get a calcium influx, it stimulates the granular migration, or insulin containing secretory granules will migrate to the cell membrane. They'll fuse with the lipid membrane, okay, and they'll secrete insulin through exocytosis, and insulin is secreted directly into the bloodstream, okay, where it can then bind and affect specific tissues like the liver. Okay, Now, this is really important kind of concept to understand because, as I said before, later on in the PowerPoint, we're going to be talking about neuronal control. So, how does our central nervous system play a role in glucose homeostasis? But of course, as I said before, there's two sources of input. Your pancreas relies on the cells themselves to kind of detect blood glucose levels, but it also relies on the autonomic nervous system, the parasympathetic and the sympathetic nervous system, to give input as well based on body blood glucose levels. So it allows for the process becomes more sophisticated and more potentiated, if you like. So just to kind of recap here, the mechanism we just discussed, our beta pancreatic cells, we've just discussed a mechanism in which they can detect glucose. And how is that? Well, passive diffusion of glucose into the cells is going to increase when you're in a fed state, because if you've had a high carbohydrate meal, you're going to be kind of closer to hyperglycaemia. Okay, so you're going to get more glucose in these cells, okay, and the ATP levels will increase. Those potassium channels that kind of contribute towards a polarized membrane, okay, will become closed, okay, and it will cause depolarization because of that increase in ATP. Why? Because glucose will undergo glycolysis. It's broken down, okay, into pyruvate, and that cycle in itself will produce a net gain of 2 ATP molecules per one glucose molecule. So if you've got a lot of glucose influx, you're going to get more ATP being produced, okay, and those potassium channels will close, and that will result in depolarization and the opening of those voltage gated calcium channels, okay, and it allows for an influx of calcium into the cell, okay, and then that stimulates granular migration, and those granules contain insulin, okay. Insulin is packaged into these granules, and calcium essentially chaperones those granules to the lipid membrane, and then the granules fuse with the lipid membrane, and you get exocytosis. The insulin is secreted into the bloodstream, and now it's free to act on tissues that express particular receptors, which we are going to talk about in the next slide. So I'll talk about this more in the next slide, but I just want to kind of introduce, just for people who aren't familiar with receptor tyrosine kinases, or RTK receptors. Insulin is a ligand for these particular receptors. Now, RTK receptors are renowned for they're famous for their tyrosine residues they have on the C-terminal, the intracellular space or compartment of this glycoprotein, if you like. And those tyrosine residues are able to phosphorylate each other, so it's autophosphorylation. And when you get ligand binding, so when insulin, for example, insulin is a ligand to these receptors, when it binds to these RTK receptors, it causes a change in the quaternary structure, and it kind of facilitates dimerization of these receptors. So two RTK receptors can come together, and then you get cross phosphorylation or autophosphorylation, and that phosphorylation results in a kind of another kind of conformational change, and that C-terminal part of the glycoprotein can act as a recruitment or docking platform for other proteins to kind of then assemble on, and then initiate a signaling cascade, an intracellular signaling cascade, which will eventually usually result in the activation of your AKT or your protein kinase B. And then obviously protein kinase B is really important in facilitating glycogenesis, which we'll look at in the next few slides. So as I said before, insulin is a ligand to these RTK receptors. So it signals through RTK receptors. Therefore, the tissues it targets will heavily express RTK receptors in their lipid membrane. So this includes your hepatocytes, your liver cells, okay, your muscle tissue, and your adipose tissue, or your fat tissue. So of course, glycogenesis, it's a process in which glucose essentially polymerizes into its polymer glycogen, okay, and glucose is stored in the form of glycogen in these tissues, okay, and the function of glycogen in these tissues is slightly different depending on where you're looking at. As I said before, in the liver, the function of glycogen is more so glucose homeostasis, so that glycogen can be stored to prevent high blood sugar levels, and of course, when blood sugar levels drop and glucagon is secreted from those alpha pancreatic cells, okay, it will facilitate glycogenolysis in the liver, which is the breakdown of glycogen into that glucose, okay, and then you have an enzyme called glucose 6-phosphatase in the liver that's expressed in the liver that will then get rid of that glucose 1-phosphate, or that phosphate group on that first carbon of the hexose ring, giving you glucose or free glucose, free to move out of the cell, okay, increasing hepatic glucose output, and raising blood sugar levels. Okay, whereas in muscle tissue, glycogen is used more so as a quick access to energy. So when you think about muscle contraction, exercise, you need that glycogen to give you quick access to glucose 1-phosphate. Glucose 1-phosphate in an ATP independent manner can be converted into glucose 6-phosphate, and then glucose 6-phosphate can undergo the glycolytic pathway and produce ATP. Of course, muscle tissue do not express the glucose 6-phosphatase for that reason, because obviously if you remove that phosphate group from the glucose, you're going to facilitate, diffusion of glucose out of those muscle cells into the bloodstream, which isn't what you want. You want that glucose to be metabolically ready to undergo glycolysis and ATP production to meet the demands of muscle contraction when you're exercising, when you're walking, when you're doing all of the above. And of course, in adipose tissue is capable of storing small amounts of glycogen, and again, that's more for quick access to glycerol. So it's not it has a slightly different functions to glucose homeostasis and slightly different functions to muscles, which is quick energy access. But either way, glycogenesis relies on insulin, okay, and glucose uptake into these cells relies on insulin signaling. So these cells, the liver tissues, your hepatocytes, your muscle cells, and your adipose tissues, your adipocytes, they all express receptor tyrosine kinase receptors, okay, and that's so insulin can signal and initiate those intracellular changes that will eventually facilitate glucose storage over glucose breakdown. Now, as I said before, insulin will bind to the extracellular alpha unit of the RTK receptor, and this results in the dimerization of the RTK receptors, okay, and you get autophosphorylation of those intracellular beta tyrosine residues, and this is referred to as ligand-induced dimerization. And this phosphorylation will create docking sites for particular protein constituents that will initiate a signaling like transduction cascade. So the two kind of once that those tyrosine residues have been phosphorylated, it creates a docking site for what's called insulin receptor substrate proteins, and their protein constituents IRS1 and IRS2, and that recruitment is really important in activating your phosphoinositide signaling mechanism, because RTK can signal through phospho or phosphatidyl or phosphatidylinositol triphosphate, that secondary messenger, which is really important in activating your protein kinase B. Now, just to take a few steps back, once we've created our docking site and our IRS1 and IRS2 constituents have been recruited, it activates phosphoinositide-free kinase. Okay, and phosphoinositide-free kinase will phosphorylate your membrane-bound phosphatidyl 4,5 bisphosphate. Now, phosphatidyl inositol 4,5 bisphosphate is a phospholipid that's expressed or found in the lipid membrane of most cells, and it can act, or it's a precursor to a really important secondary messenger. Now, phosphoinositide free kinase will phosphorylate phosphatidyl 4,5 bisphosphate, okay, giving it an extra phosphate group on its hydroxyl group, okay, becoming phosphatidyl inositol 3,4,5 triphosphate. And that phosphatidyl 3,4,5 triphosphate is really important in spatial recruitment, okay. A lot of proteins that have what's called a plextrin homology domain can respond to PIP3 formation. So this usually happens on quite a large scale. There's a lot of PIP2, if you like, so phosphatidyl inositol 4,5 bisphosphate throughout the lipid bilayer, especially in close proximity to these RTK receptors. And when you get insulin binding, okay, and phosphoinositide free kinase activation, you're going to get a lot of phosphorylation of that phosphatidyl inositol 4,5 bisphosphate to phosphatidyl inositol 3,4,5 triphosphate. That's going to cause a change in pH in those areas in the cell membrane, and it's going to enable certain proteins to migrate towards the cell membrane. So one of those proteins is your protein kinase B, and another one of those proteins is your 3-phosphatidylinositide protein dependent serine threonine kinase 1. Okay, so this is really important in this is a really important step, because we want to activate protein kinase B, and in order to do that we need our PDK1. But PDK1 and AKT, or protein kinase B, aren't in close proximity to each other. But a way that you can get them, the way you can kind of facilitate spatial recruitment, is through phosphatidylinositol 3,4,5 triphosphate, or that kind of lipid secondary messenger, acting as that kind of pH, that kind of a pH a change, if you like, stimulating that kind of migration or spatial recruitment of those proteins, because then your PDK1, or your phosphoinositide protein dependent kinase, is able to phosphorylate AKT, or protein kinase B, and activate it. Now, once you get the activation of your protein kinase B, what you get is a series of kind of phosphorylation reactions, if you like. Covalent phosphorylation, which, as you know, is really important in regulatory biology. Phosphate groups are removed and added to regulate proteins, activate and inactivate them accordingly. So protein kinase B is really important in facilitating glycogenesis, because it inactivates or phosphorylates enzymes like glycogen synthase kinase 3. Now, glycogen synthase kinase 3 is an enzyme that will phosphorylate glycogen synthase, keeping it in its active form. But when glycogen synthase kinase 3 becomes phosphorylated, it's unable to phosphorylate glycogen synthase. Okay, so you get less glycogen synthase inhibition, and glycogen synthase is a really important enzyme in glycogenesis. Now, protein kinase B also stimulates the activation of protein phosphatase 1. Now, protein phosphatase 1 is really important because, as I mentioned before, our glycogen synthase kinase 3 will phosphorylate glycogen synthase. In order to get glycogen synthase active, you need to remove that phosphate group, and that's where protein phosphatase 1 comes in. Okay, so when protein phosphatase 1 removes those phosphate groups from glycogen synthase, it kind of extenuates its activity to prevent it kind of going back on itself. Because if you think about it, if glycogen synthase kinase 3 has been inactivated, there may still be some phosphorylated glycogen synthase that we need to dephosphorylate. So we need protein phosphatase to do that. Now, protein kinase B will also stimulate GLUT4 translocation to the lipid membrane, especially in the liver. Now, this allows for increased glucose uptake. As you know, glucose goes from the bloodstream and it can passively diffuse into cells through GLUT transporters. And in the hepatocytes, it's GLUT4, because you get an increase in glucose uptake. And obviously, we need that glucose uptake to facilitate our glycogenesis. But before we move on, I want to touch more on lipid secondary messengers and signaling. I know that I haven't really spoken about cell signaling, but because we're talking about hormonal control and regulation, I think it's important to understand what's actually happening. So you can see I've got a diagram of the plasma membrane, which is made up of several phospholipids. And one of those phospholipids is your phosphatidylinositol 4,5 bisphosphate. And usually phosphatidylinositol 4,5 bisphosphate will be in close proximity, or in high concentrations close to receptor tyrosine kinase proteins or glycoproteins. So when you get ligand binding of insulin, for example, and you get the activation of your phosphoinositide 3 kinase, it can then work on its substrate, its phosphatidylinositol 4,5 bisphosphate. It can phosphorylate that into your phosphatidylinositol 3,4,5 triphosphate. Okay, and when you get a lot of conversion of PIP2 into PIP3, or phosphatidyl 3,4,5 triphosphate in the lipid membrane, you get a pH change, and that pH change facilitates a spatial recruitment for two particular proteins that are really important in activating protein kinase B. So the first one, of course, is your PDK1, and the second one is called the mTORC2, which I didn't mention in the previous slide. Now, mTORC2 is a multi-protein kinase complex. Okay, and it stands for mechanistic target of rapamycin complex 2. And again, because it's a kinase, it phosphorylates. So protein kinase B, or AKT, requires activation through two separate entities, or two separate enzymes, that being PDK1 and mTORC. Now, both PDK1 and AKT or protein kinase B have what's called plextrin homology domains, and they're domains that are able to respond to phosphatidylinositol 4,5 bisphosphate conversion into phosphatidylinositol 3,4,5 triphosphate conversion, that pH change. There's domains in those proteins that are very sensitive to that pH change, and it facilitates what's called spatial recruitment. So typically in a cell, when you have high levels of PIP2 before you have insulin binding, you're going to have the protein kinase B, the mTOR-C complex, and the PDK1 enzyme all not in close proximity with each other, so they can't activate protein kinase B. But of course, when they're in close proximity to protein kinase B, and of course when protein kinase B has also migrated to that plasma membrane, that PDK1 and your mTOR-C 2 complex is able to phosphorylate your protein kinase B and activate it. Now, as I said before, it's really important both entities are required to activate this protein kinase B. So the mTOR complex will phosphorylate the serine domain of the protein kinase B, because remember, kinases have serine threonine domains. That's really important, integral to their function. So the mTOR-C2 complex will phosphorylate that serine domain of the protein kinase B, whereas your phosphoinositide protein dependent kinase 1 will phosphorylate the threonine of the protein kinase B. Okay, and once the serine has been phosphorylated by your mTOR complex and the threonine has been phosphorylated by your phosphoinositide protein-dependent serine threonine kinase 1 enzyme, you get an active protein kinase B. And then protein kinase B, as I mentioned before, will facilitate or activate protein phosphatase 1, which will help with glycogen synthase activity, and protein kinase B will also inhibit enzymes like glycogen synthase kinase 3, preventing further inactivation of glycogen synthase. And of course, glycogen synthase is one of the enzymes that we need to facilitate glycogenesis. Now, I think it's really important to kind of acknowledge as well that insulin binding results in a transcriptional shift in cellular processing, as does with most signaling. That intracellular, that signal transduction pathway results in many changes in the cellular environment. Okay, and it will shift genes, and you'll see there's a pattern between glucagon and insulin signaling. Glucagon is the antithesis of insulin signaling, and I think that's very safe to say from a scientific perspective, because insulin, when it binds to these cells, it's facilitating glucose storage and glycogenesis, but it's also facilitating glycolysis, and because it's facilitating glycolysis and glucose breakdown, you're going to get increased pyruvate and acetyl-CoA production. So therefore, you're going to need to facilitate lipogenesis in the liver as well to deal with that pyruvate and acetyl-CoA. And of course, glycogen is a great storage molecule, okay, but it has its limits, which I'll talk about later, which is why you need fatty acid or fat storage as well. So insulin will facilitate many changes in the cell, but it will also inhibit certain metabolic processes that are counterintuitive. For example, glycogenolysis, the breakdown of glycogen. If we're trying to facilitate glucose storage, it doesn't make sense to have glycogenolysis running at the same time. It will completely disrupt our physiology, because we're trying to lower blood sugar levels, not increase them. Okay, and another example would be gluconeogenesis, so the synthesis of glucose from amino acids. Of course, insulin's goal is to lower blood sugar levels, so it's going to inhibit gluconeogenic gene expression. Okay, it's going to inhibit beta oxidation, the breakdown of fats, but it's going to facilitate the storage of glucose, it's going to facilitate glycogenesis, and it's going to facilitate or upregulate the expression of lipogenic genes like your acetyl-CoA carboxylase, which I'm all going to get into as we kind of move through these slides. But the main takeaway from this slide, I guess, is to understand the importance in spatial recruitment to the plasma membrane that is essentially induced by our PIP3 or our phosphatidylinositol 3,4,5 triphosphate. That change in pH will stimulate migration of your mTOR-C2, your PDK1, and your AKT or protein kinase B. It will bring them all in close proximity to each other in the plasma membrane, okay, and it will allow for your PDK1 and your mTOR-C2 to activate protein kinase A through phosphorylation. Okay, it has to be phosphorylated. It's threonine and it's serine to be domains to become activated, and of course those plextrin homology domains in the diagram that I've got in blue are the domains in the proteins that allow them to respond to PIP3 synthesis, or PIP3 conversion, if you like. And of course, what's the enzyme that will convert PIP2 into PIP3 is our phosphoinositide kinase, and that's activated by our insulin binding to the RTK receptors. So I've got this signaling transduction cascade along with the enzyme activation all here, just to kind of recap so we can kind of understand what we're talking about. So first of all, insulin is secreted via the beta pancreatic cells. And how does that happen? Okay, so we have nerve input from the hypothalamus, from homeostatic centers in the hypothalamus that can detect ATP and glucose levels. But then we also our beta pancreatic cells are also capable of sensing glucose levels and responding to elevated glucose levels directly. And how do they do this? So they've got GLUT2 transporters in their lipid membranes of these pancreatic cells, allow for the passive diffusion of glucose into these cells. Increased glucose results in increased glycolysis. You get an increase in ATP. You get a change or a fluctuation in your adenosine triphosphate to adenosine diphosphate ratio. These beta pancreatic cells also have voltage gated potassium channels in their lipid membrane. Okay, and when ATP levels increase, these channels close and it causes depolarization. Okay, and that depolarization allows or stimulates the opening of calcium gated ion channels that are also expressed in the lipid membranes of these beta pancreatic cells. Okay, it allows for an intracellular influx of calcium. Okay, and that calcium stimulates cell-mediated exocytosis of insulin, so it causes granular migration. Those granules containing insulin will migrate to the lipid membrane of these beta pancreatic cells. They'll fuse with that lipid membrane and they will secrete insulin directly into the bloodstream, and calcium is acting as that chaperone, if you like. Now, once insulin is secreted into the bloodstream, we're going to mainly focus on the liver here because we're talking about glucose homeostasis. Liver tissues, like your hepatocytes, in their lipid membranes, they express high densities of receptor tyrosine kinase, glycoproteins, and they also, in close proximity, they also have a lot of phosphatidylate 4,5 bisphosphate phospholipid in close proximity to those RTK receptors. Once insulin binds to those RTK receptors, you get dimerization and autophosphorylation of those tyrosine residues. Okay, those two RTK receptors dimerize, and those tyrosine residues will phosphorylate each other, and this creates a docking platform, if you like, and it allows our IRS1 and IRS2 protein constituents to be recruited, and this results in the activation of our phosphoinositide 3 kinase enzyme. And that phosphoinositide 3 kinase enzyme is able to phosphorylate the membrane-bound phospholipid phosphatidyl 4,5 bisphosphate into phosphatidylinositol 3,4,5 triphosphate. When that happens on a large scale, when you get a lot of PIP3 conversion, or PIP3 conversion from PIP2, you get a change in pH, and this is integral in facilitating the spatial recruitment of your PDK1, protein kinase B, and mTORC2 enzymes. Okay, and both PDK1 and mTORC2 are required to activate protein kinase B through phosphorylation. PDK1 and protein kinase B possess those plextrin homology domains, which allow them to respond to the increase in phosphatidylinositol 3,4,5 triphosphate concentration. Okay, they migrate towards the plasma membrane. They're in close proximity to each other, so they're able to phosphorylate. PDK is able to phosphorylate protein kinase B. Okay, and mTORC2 is able to phosphorylate protein kinase B. Protein kinase B then becomes activated. Now, protein kinase B is able to phosphorylate glycogen synthase kinase 3, or GSK3, and glycogen synthase kinase 3 typically will inhibit glycogen synthase through phosphorylation, but of course, when it itself becomes phosphorylated by protein kinase B, is inactive and is unable to do that. Okay, and protein kinase B will also lead to the activation of protein phosphatase 1. Now, once glycogen synthase kinase 3, before it was phosphorylated by protein kinase B, it was phosphorylating a lot of glycogen synthases. Okay, and there's a lot of inactive glycogen synthases, and all that needs to be done to make them active again is to remove that phosphate group, and that's where our protein phosphatase 1 comes in. It will dephosphorylate glycogen synthase, further kind of facilitating its activation. And of course it can't be inactivated again because our glycogen synthase kinase 3 has become inhibited through phosphorylation by protein kinase B. Now we have our glycogen synthase active, or in a more active state. Okay, we can now facilitate glycogenesis more and glucose storage, and of course protein kinase B will also stimulate GLUT4 translocation to the hepatocyte lipid bilayer, okay, increasing glucose uptake into those cells. Now, insulin actively kind of facilitates glycolysis, so the breakdown of glucose into pyruvate, okay, and you get the ATP production there. Now, that's obviously really important because a lot of that glucose that isn't being used for storage will be used for direct, ATP access through those cells. Like we're talking about the liver here mainly because we're talking about glucose homeostasis, and of course, the liver plays a really important role in kind of managing or maintaining blood glucose levels. So I mentioned before that insulin signaling will induce transcriptional changes, okay, changes in transcriptional processing. So you'll get a shift in gene expression because of insulin signaling. So insulin will kind of will facilitate anabolic processes. So glycogenesis is an anabolic process. Glucose is polymerized into glycogen. Similarly, as I mentioned before, it makes sense that glycolysis is upregulated from an evolutionary perspective. A lot of that glucose, we don't want too much glucose in the cell because that could become toxic. So a lot of that glucose is also broken down, okay, into pyruvate. And of course, we don't want too much pyruvate in a cell either. And glycogen, as I mentioned before, has its limits when it comes to storage. So a lot of that glucose will be indirectly stored as fat. So it's converted into fat through an enzyme called acetyl-CoA carboxylase, which will convert acetyl-CoA into malonyl-CoA, and that's a rate-limiting step in fatty acid synthesis. Okay, the synthesis of fatty acids to be stored. So therefore, insulin will induce transcriptional changes that will facilitate glucose storage and lipogenesis, so the synthesis of fatty acids. Now, as I mentioned before, there's transcriptional shifts, and this is mainly to do with protein kinase B. It phosphorylates transcription factors, suppressing gluconeogenic genes. So FOXO1 is a transcription factor that is known for promoting the transcription of gluconeogenic enzymes and glucose 6-phosphatase as well. Now, glucose 6-phosphatase in the liver, as I mentioned before, will remove that glucose, that phosphate group from the glucose, allowing for it to diffuse out of the cell. But of course, we're trying to lower blood sugar levels, so we don't want hepatic glucose output. So when insulin binds to its RTK receptors and that transcriptional shift happens, that signal transduction cascade results in a transcriptional shift, okay, and it will facilitate more glucose storage. So enzymes like glucose 6-phosphatase will be suppressed to prevent that, prevent glucose from becoming dephosphorylated. It will also suppress gluconeogenic genes, of course, because we don't need more glucose. And of course, gluconeogenesis is the process in which amino acids are used to synthesize glucose, mainly alanine, like glucogenic amino acids. And there's certain enzymes like PepCK that are really important in this process. So inhibiting them will prevent gluconeogenesis. Now, of course, we want lipogenesis, as I mentioned before, because we've got an increase in glycolysis. So naturally, we're going to have more pyruvate and then acetyl-CoA expression because more cellular respiration is happening. And of course, to prevent ATP toxicity and also to prevent a buildup of all these intermediates, another option a cell has is to store some of that pyruvate and that acetyl-CoA. Of course, that pyruvate is converted into your acetyl-CoA. Then your acetyl-CoA carboxylase converts it into malonyl-CoA, which will initiate fatty acid synthesis. Of course, beta oxidation is also inhibited upon insulin binding. Insulin inhibits beta oxidation because insulin is mainly anabolic. Okay, lipogenesis is an anabolic process, whereas beta oxidation is catabolic. It's counterintuitive. If you are stimulating beta oxidation, okay, then you're essentially going back on yourself because we're trying to build up fats for storage from these carbohydrates. If beta oxidation is happening in these cells, then it's going to be counterintuitive. The same way if glycogenolysis is happening in these cells, it's going to be counterproductive to our glycogenesis. Okay, we want to build that glycogen and we want to build those fats. So beta oxidation is suppressed, and that's typically through malonyl-CoA production. Malonyl-CoA prevents fatty acid transport into the mitochondrial matrix where beta oxidation takes place. Now, as I said before, insulin is mainly associated with anabolic processes. So glucagon, being the antithesis of insulin, which we'll talk about later, is associated more with catabolic processes. But of course, not all the time, because when you look at gluconeogenesis, right, that's more anabolic. You're building glucose from those amino acids. So now we're going to talk more about glycogenesis, because this is a really important metabolic process, and it's a polymerization process. And it's not, in my opinion, it's not too complicated to understand, and it's quite hard to visualize when you don't have a whiteboard and a pen. So I'll do my best to try and explain it. But glycogen, or glycogenesis, is a synthesis of glycogen from glucose. Now, as you know, glycogen is a polymer. It's made up of many repeating monomeric subunits, and the subunits are glucose. So it's a polysaccharide made up of many monosaccharides, because glucose is a monosaccharide. Now, glycogenesis occurs during a fed state because it prevents hyperglycaemia. A lot of glucose needs to be stored in the form of glycogen. Of course, the liver can take up glucose and it can undergo glycolysis, but we can't have too much ATP being produced in cells, and we can't have a buildup of all these metabolic intermediates. So glycogenesis is integral because it allows for glucose storage. And then, of course, if you're storing that glucose, it can then be used later. So that when blood sugar levels drop later on in the day, when you're in a fasted state, that glycogen can be broken down in the liver through glycogenolysis into its glucose 1 phosphate, and then that glucose 6 phosphatase can then get rid of that phosphate group and then give you free glucose to allow for hepatic glucose output back into the bloodstream. But anyway, back to glycogenesis. So of course, as people know, what you're looking at here is a glucose molecule. Now, glycogenesis is not as simple as this. A lot of people remember, when you're looking at the very basic kind of building blocks of biological molecules, you look at condensation reactions that occur between sugars. So glucose is able to bond, right, mainly through alpha 1 to 4 glycosidic bonds. So the first carbon, that non-reducing end on the the reducing end on that first glucose molecule, combined to the non-reducing end, the fourth carbon of the other glucose molecule, and water is given off. Okay, and that's a glycosidic bond, an oxygen bridge. And of course, glycogen has a glycosidic bond, but the polymerization process is a little bit more complicated than what's being shown here, because it involves activation through, phosphorylation, as most processes in biology do. So what you're looking at here is a very kind of simplistic, just to kind of understand glucose as a single sugar. Okay, so it's a hexose sugar, it's got a 6-carbon ring, and you have those hydroxyl groups, and those hydroxyl groups are really important in chemistry behind, condensation reactions and forming those glycosidic bonds. So I mentioned before, the polymerization process is not as simple as what was shown in the previous slide. That's not really polymerization. That's just the formation of a disaccharide from a monosaccharide. You can either use the first carbon and bond it with the the fourth carbon on an adjacent glucose molecule, or the first carbon to the 6th carbon on another glucose molecule to form an alpha 1 to 6 glycosidic bond. And glycogen contains both alpha 1 to 4 and alpha 1 to 6 glycosidic bonds, and those alpha 1 to 6 glycosidic bonds cause branching. So glycogen is not just like a linear polymer, or like DNA as a polynucleotide. It's a branch chain polymer, so it's got lots of branches, and that's because of the 1 to 6 glycosidic bonds, as well as the alpha 1 to 4, which I'll show in a diagram in a slide later on. But as I mentioned before, like with most metabolic processes, we need leaving groups to activate our metabolites. So this phosphorylation of glucose to glucose 6 phosphate is a really important step. Okay, and this is done by either glucokinase or hexokinase, depending on what tissue you're looking at. So glucokinase or hexokinase will transfer a phosphate group using ATP or adenosine triphosphate as that phosphate donor. Like in most cases in biology, it will take that phosphate group and it transfers it to the 6th carbon on the hexose ring. Hence, you get glucose 6 phosphate. Now, glucose 6 phosphate is essentially a metabolic hub. When we looked at the hexomonophosphate pathway, we looked at how glucose 6 phosphate serves as a metabolic hub and has many different fates in metabolism, depending on cellular needs and depending on where you are in the body. Now, we won't go over it, but just to kind of recap, glucose 6 phosphate can either be used for glycolysis, so it can be further broken down into pyruvate, or glucose 6-phosphate can be shunted to the pentose phosphate pathway. It can be oxidized into phosphogluconolactone, then it's committed to the hexamonophosphate shunt. It can become ribose 5-phosphate, and then it can either feed back into glycolysis, or it become phosphoribosyl pyrophosphate, or PRPP, and then it gets fully committed to becoming a ribose sugar that you see in your nucleic acids like your RNA or your DNA. And another fate glucose 6-phosphate has is glycogenesis. So you need phosphorylated glucose to polymerize it, but you don't need glucose 6-phosphate. What you actually need is glucose 1-phosphate. So you can see once glucose 6-phosphate is converted into glucose 1-phosphate via phosphoglucomutase, which is able to transfer that phosphate group in an ATP independent manner. That's really important because it's a very efficient process. Okay. In an ATP independent manner, okay, you get glucose 1-phosphate, then we're essentially committed to glycogenesis. But the polymerization process actually requires more activation of this glucose molecule, and how we do that is by adding on a UDP group, so a uridine diphosphate group. So UTP is used, or uridine triphosphate is used as our donor, if you like, our UDP donor. Okay, and UDP glucose pyrophosphorylase 2, or UGP2, is the enzyme that will get rid of that that gamma phosphate on the UTP to give us UDP, and it attaches it to that glucose molecule, giving us UDP glucose. And UDP glucose is really important in this polymerization process. Okay, it's a really good leaving group, and it allows for that polymerization. But the first step of glycogenesis, the first real kind of step of glycogenesis isn't actually enzyme controlled. It's called autoglucosylation. Okay, so this is happening. This happens automatically without enzymes. The first few residues that kind of build, but we need a primer, kind of like with DNA synthesis and RNA. You need RNA as a primer to then elongate and build DNA using that RNA primer. Okay, it's the same with glycogen. Okay, you need a primer first, and that glycogenin kind of acts as a pre-primer, if you like. Okay, it allows for autoglucosylation, and it allows for UDP glucose to bind to this glycogenin, okay, giving off its UDP, forming short residues of alpha 1 to 4 glycosidic bonds, oligomer chains of short glucoses with alpha 1 to 4 glycosidic bonds. Now, glycogenin is a protein, so when you look at glycogen, when it's stored in the center of its structure, you see the glycogenin, and then coming out of it, you've got the glycogen chains, if you like, and then you've got the branches. So that's kind of how it's stored, and I'll show that in the coming slides. Now, glycogenin has a lot of tyrosine. Now, tyrosine has a phenyl or phenyl ring, and that kind of alcohol ring, and that hydroxy group, which is really good at acting as a nucleophile. So it allows for glucose to bind to that glycogenin, okay, and that UDP group is a leaving group, so it's removed, it's given off, and the tyrosines kind of facilitates that autoglucosylation or glycosylation, and you get a bond between glucose and glycogenin to start the whole thing off, and that glucose will then accept another glucose residue, okay, so it will form a 1 to 4 glycosidic bond, and you get residues kind of coming out of this glycogenin that are about 8 residues in length, 8 glucose molecules in length, and it's a linear oligomer, so we haven't formed our alpha 1 to 6 branches yet. So you can kind of visualize now what glycogen looks like. You can see the glycogenin in the center of the structure. That's that protein that's rich in tyrosine, okay? Those phenyl rings in tyrosine, in the tyrosine R group, have that hydroxyl group, which is really good nucleophile, and it allows for that kind of initial autoglycosylation. Now, the enzyme glycogen synthase kind of comes in once we've got these primers, these short kind of alpha 1 to 4 glycosidic bond primers that are about 8 residues in length. Glycogen synthase is able to extend these. It can transfer UDP glucose directly onto this growing branch. Okay, it's like a form of elongation. And then you have a glycogen branching enzyme called amylo 1 to 4 1 to 6 transglucosylase. Okay, and that transfers residues. It transfers 6 to 7 glucosal residues from one oligomer chain to a nearby hydroxyl group, a C6 hydroxyl group on another chain, creating an alpha 1 to 6 branch point. So it allows for more storage. Okay, it increases its solubility, and it multiplies the number of reducing ends. So it allows for more monomeric subunits of glucose to essentially be put on to this glycogen, this large glycogen polymer. And it's a really important process, and that glycogenin remains in the center, okay, because that glycogenin is integral in starting off this process of glycogenesis. But the formation of those first few primers isn't enzymatically controlled. It happens because of the chemical nature of tyrosine. That hydroxyl group in the phenyl ring is a really good nucleophile, and it allows for that autoglycosylation. And then once the branch gets sufficiently long, that's when glycogen synthase is able to directly extend that growing polymer, and then your transglucosylase enzyme is able to essentially take a few residues from those chains and then put them onto the hydroxyl group on a CIF carbon of another glucose molecule on a growing oligomer stemming from another portion of the glycogenin, because glycogenin has tyrosine all on its outer kind of layer of that kind of it's like a globular protein that's got loads of tyrosine on that on the kind of outer layers, allowing for points for glucose or UDP glucose to come in there and also glycosylate. And of course, UDP is given off in this chemical reaction. If we're doing it as a chemical reaction, which typically most courses you don't really need to know it as a chemical reaction, you just need to understand the concept of branching and storage, and most of the time you don't actually need to know the mechanisms behind glycogenesis. So we just kind of looked at how insulin kind of facilitates glucose storage, okay, and how our bodies deal with hyperglycaemia, so when blood sugar levels raise when you're in a fed state. But now we're going to look at how our body deals with hypoglycemia, or lower blood sugar levels, when we're in a fasted state. Okay, so this is when you've gone a long time without eating carbohydrates, you've gone a long time without eating food in general. Okay, or for whatever reason your blood sugar levels have dropped, and that could be because of your diet. So again, if you're on an Atkins style high protein low carb diet, then your blood sugar levels are going to be lower. So glucagon is going to be secreted by these kind of alpha pancreatic cells in response to that. Now, I'm going to talk about this in a lot more detail as the slides go on, but I'm just going to kind of summarize the process so you can kind of appreciate how it kind of is the opposite of insulin and glycogenesis, how glucagon and glycogenolysis kind of facilitates and helps kind of push us towards increasing glucose in the blood rather than getting rid of glucose from the blood. So your pancreas, as I said, your islets of Langerhans, that tissue contains alpha cells as well as beta pancreatic cells, and the alpha cells are responsible for synthesizing and secreting glucagon. Again, glucagon is a peptide hormone, so it's synthesized through transcription and translation, okay, as opposed to, say, cholesterol synthesis from cholesterol, because it's not a steroid hormone. Now, when talking about glycogenolysis, glycogenesis, of course, it happens in your muscle cells. Glycogenesis happens in your liver cells, and glycogenesis also happens in your adipose cells, your fat cells. But glycogenolysis happens in all of the above, but it serves different purposes. Glycogenolysis in muscle cells is to have quick access to glucose 6-phosphate for cellular respiration, ATP production, to meet demands for muscle contraction, because muscle contraction is heavily ATP dependent process. Now, glycogenolysis in the liver serves slightly different to glycogenolysis in the muscle. So, as I said before, the liver is for glucose homeostasis, one of its functions, one of its many, many important functions. Now, liver cells express glucose 6-phosphatase. And that's really important, because once glycogenolysis is glycogen's broken down, as you know, it's polymerized through phosphorylation and UDP for that leaving group. Well, it's also broken down through phosphorylysis, okay, so adding inorganic phosphate. to break those glycosidic bonds and to give you glucose 1-phosphate. And of course, when you get glucose 1-phosphate in your hepatocytes, your phosphoglucomutase in an ATP independent manner can transition that phosphate group to the 6th carbon on the hexose ring, giving you glucose 6-phosphate. And then glucose 6-phosphatase can come along, remove that phosphate group, giving you free glucose, and that is able to leave the cell through those GLUT4 transporters into the blood, increasing hepatic glucose output, and you get, your blood sugar levels are rising. Now, glucagon is the hormone secreted by those alpha pancreatic cells that will facilitate and stimulate this process through signal transduction mechanisms, which we'll go over in more detail as we go on through the slides. Now, we mentioned before insulin binds to the RTK receptors. Insulin's a ligand of the RTK receptors. Well, glucagon is a ligand of the G protein couple receptors. Now, G protein couple receptors are those kind of large 7-domain transmembrane receptors that have that heterotetrameric or heterotrimeric protein subunit, and they act as kind of like guanosine exchange factors, if you like, or guanine exchange factors, and they eventually activate your adenylyl or adenylate cyclase. And adenylate cyclase can then produce high levels of the secondary messenger cyclic adenosine monophosphate. And as a lot of people should be familiar with, your cyclic adenosine monophosphate is really important in activating protein kinase A, and protein kinase A is really important in facilitating glycogenolysis, the same way protein kinase B is really important in facilitating glycogenesis during insulin signaling. So you can kind of start to compare these processes in terms of their signal transduction mechanisms. Now, as I mentioned before, the liver is responsible for glucose homeostasis, okay, and glycogenolysis is initiated through glucagon signaling through G protein couple receptors in the liver. Now, in muscle cells, it's slightly different. In muscle cells, it's signaled through adrenaline, okay, so epinephrine. Okay, adrenergic receptors. Okay, it's a slightly different signaling transduction mechanism, but glycogenolysis is also initiated in muscle cells through calcium binding to calmodulin, and that's really important because, as a lot of people know, muscle contraction is heavily dependent on calcium ions. So we'll look at how glycogenolysis is regulated in muscle cells later, but just to kind of be aware, there are slight differences in the way glycogenolysis happens in both muscle cells and liver cells. And of course, in our fat cells, we're not really going to be talking about that today, but glycogenolysis is really important because it allows those cells to have access to glycerol. So, as I said before, our G protein couple receptors are really important in glycogenolysis in the liver. Okay, they activate our cyclic adenosine monophosphate, which activates our protein kinase A. Okay, and will facilitate glycogenolysis. It will also activate gluconeogenic genes, of course, because we want to increase our glucose overall. So amino acids can be used to make glucose as well, and this is really important actually, because when you think about people who might be on high protein low carbohydrate diets, okay, glucagon isn't just released through low blood sugar levels. It can also be released, or its release can be stimulated due to raised levels of amino acids. So if you're on a high protein diet, then those amino acids can then be used to make to make glucose, glucogenic amino acids in particular, and glucagon can facilitate that process by activating gluconeogenic genes like your PEP-CK and your glucose 6-phosphatase. But before we expand on that, I'm going to take a few steps back and we'll look at glucagon. So as I said before, glucagon is a 29 amino acid peptide hormone. So it's a polypeptide composed of 29 amino acids. Okay, and it's obviously, because it's a peptide hormone, it's synthesized during transcription and translation, the same way insulin is, only it's synthesized in those alpha cells. Okay, it's synthesized and secreted in those alpha cells in response to hypoglycaemia. So lower blood sugar levels, but it can also be secreted in response to elevated amino acid levels. And of course that makes sense. As I said before, a lot of people who want to be on high protein low carb diets for weight loss, or if they're trying to if that works for them, if that's healthier for them, they might choose that option. And of course, us being omnivores, and our metabolism, we're very flexible, we're not like obligate carnivores and we're not like herbivores, we have that option to be able to generate energy and uses from all sorts of foods. So if you're on a high protein diet, okay, you've got elevated amino acids, your blood sugar level is going to be lower. So what your liver is able to do is able to use those amino acids, those glucogenic amino acids like alanine, to essentially synthesize glucose, okay, and it kind of converts it into Krebs cycle intermediates like oxaloacetate, and eventually into your pyruvate, and then it can essentially reverse glycolysis, which I'll go over in another video. And of course, then that glucose 6-phosphatase can get rid of that glucose 6-phosphate into free glucose and allow for glucose to be distributed into the blood to tissues that need it. And of course, that's when you're on a high protein diet. And of course, it's the same principle. If you have hypoglycaemia, of course, then a lot of that glycogen that's stored in the liver already can be broken down into glucose 1-phosphate. Phosphoglucomutase can convert that into glucose 6-phosphate, and then glucose 6-phosphatase can convert that into free glucose to leave the cell and enter the bloodstream to raise blood sugar levels, increase hepatic glucose output. Now, I will also talk about more transcriptional changes as we kind of move on, but just to kind of understand, just so that everyone's kind of following me, glucagon will promote glycogenolysis, okay, and raise blood sugar levels, whereas insulin will promote glycogenesis and facilitate glucose storage and lower blood sugar levels, okay, and they're both secreted in response to different blood sugar levels. So insulin is secreted in response to hyperglycemia, and glucagon is secreted in response to hypoglycemia. But of course, in an exam, and they might want to kind of make this distinction, glucagon is also secreted in response to elevated amino acids in the blood. And of course, it makes sense. Just think about Atkins diet, think about paleo diets that are low in carbohydrates. Of course, in all cells, even in obligate carnivores like lions and tigers, glucose is the primary source for ATP generation, okay? Cellular respiration is a core process in all eukaryotic organisms, okay, even obligate carnivores. Only obligate carnivores rely on gluconeogenesis to create glucose from those amino acids to then be used for cellular respiration, whereas with us, gluconeogenesis is more of an option in either when you're in a fasted state, okay, when cortisol and glucagon are being released, or it's an option when we're having a high amino acid diet and we're having low carbohydrates. And I'm not saying one's worse than the other, I'm just saying our cells are actually very flexible, so it allows us to have high protein meals. Some days you can have high carbs and low protein, some days you can have a lot of fat, okay. It just depends on what you're feeling, and it's because of our physiology and our very complex biochemistry that we're able to do that, and we're able to make use of all these different kind of food sources. So as I said before, glucagon is secreted by those alpha cells, and they kind of neighbour beta pancreatic cells in the islets of Langerhans, along with delta cells. They kind of make up that tissue, and this tissue is really important in regulating blood glucose levels. It can do it directly and intrinsically through ATP, of course, because we know the relationship between cellular glucose concentrations and ATP levels and ATP to ADP ratio. But of course, they also rely on autonomic nerve input from the sympathetic and parasympathetic nervous system as well to potentiate and amplify either glucagon release, insulin release, glucagon suppression, or insulin suppression, depending on what our blood sugar levels are doing. And of course, these alpha cells in the islets of Langerhans can also detect amino acid concentrations in the blood as well. So I'm going to talk about first of all, I'm going to talk about how glucagon secreted through this kind of direct sensing of blood glucose levels, and then we're going to talk about paracrine inhibition, which plays a really big role in glucagon secretion as well. So those pancreatic alpha cells act as metabolic sensors. So glucagon can be secreted through these kind of glucagon can be secreted through this kind of exocytosis, again through granular migration. Glucagon is contained in these granules. Now, when ATP is low, of course, that's when there's not a lot of glucose, okay, because there's not a lot of glycolysis happening. Not a lot of glucose can go into these cells, okay, so ATP levels are going to fall. Those ATP sensitive channels, again, they will close, okay, and there will be depolarization, okay, and this allows for the opening of these sodium gated and calcium gated ion channels, okay, and it allows for that calcium influx, and it facilitates glucagon-mediated exocytosis or cell-mediated exocytosis of glucagon. And it's a similar principle to the beta pancreatic cells. The only difference is the beta pancreatic cells are responding to elevated ATP, whereas the depolarization in our alpha cells are responding to decrease in ATP production, which is to be expected if you have less glucose coming into the cell. So I mentioned you have the pancreatic islets. You've got they're composed of the beta cells which secrete insulin, okay, the delta cells which secrete somatostatin, and the alpha cells which secrete glucagon. But of course, the beta cells also secrete gamma aminobutyric acid. They also secrete somatostatin, okay, and they secrete insulin, and that has an effect on the alpha cell and glucagon synthesis and secretion. And so does the delta cell, because the delta cell will secrete somatostatin. So when you're in a hyperglycemic state, okay, beta cells will not just secrete insulin, but they'll also secrete zinc and GABA, okay, and your delta cells will secrete somatostatin, and beta cells will also secrete somatostatin during hyperglycemic conditions. So those somatostatin will bind to G protein couple receptors on your alpha pancreatic cells, okay, and it reduces cyclic adenosine monophosphate production, okay, because it's the GI protein couple receptors which inhibit adenylate cyclase, okay, not the GS or the GQ, it's the GI ones. So the GI are the inhibitory ones that result in a decrease in intracellular secondary messenger cyclic adenosine monophosphate, and that will inhibit that influx of calcium, and it will prevent a granular migration of glucagon, okay, and it will prevent glucagon release. So GABA will also hyperpolarize a cell, okay, and it will prevent the opening of those voltage-gated calcium channels, because those voltage-gated calcium channels only happen when there's depolarization. Okay, if there's hyperpolarization and there's too much potassium in those cells, then there's not going to be an influx of calcium. Okay, so glucagon will not be released. Okay, and GABA is released in hyperglycemic conditions as well. So this essentially means that glucagon secretion is much more governed by insulin secretion than vice versa. So hyperglycemic conditions will prevent glucagon secretion not just through insulin, but through GABA, zinc, and somatostatin. And your delta cells also release somatostatin too. And as I said, that somatostatin binds to your GI protein couple receptors, and it actually inhibits the adenylyl or adenylate cyclase enzyme, that membrane-bound enzyme, from producing a lot of that CAMP from ATP. So during hypoglycemia, however, insulin and that somatostatin drops dramatically, okay, and so that calcium influx can be facilitated, if you like, and it can then allow for glucagon secretion or cell-mediated exocytosis of glucagon into the bloodstream. So glucagon signals through G protein coupled receptors. And I mentioned before, muscle cells do not have glucagon receptors. To initiate glycogenolysis, they rely on epinephrine or adrenaline, but adrenaline signals through alpha-1 adrenergic and beta-2 adrenergic receptors, which are respectively GQ and GS coupled protein receptors. So they're still metabotropic G receptors, but the ligand, or the respective ligand that's initiating that signal transduction, is different in muscle tissue as it is in liver tissue. And I think this diagram is really good because the GS and GQ protein transduction pathways are pretty much identical in muscle tissue to liver tissue. The only difference is obviously the intracellular effects, but also the ligand that's binding to that receptor. So I think this is a really good diagram, and I'll just kind of talk everybody through it before we kind of move on to the next slides, and I'll go over it in more detail. And for those people who are not familiar with metabotropic G protein receptors, I'll go over them in more detail in another video. But essentially, G protein couple receptors, they're a family of receptors that rely on guanosine triphosphate, or GTP, to activate them, and that happens, that guanosine exchange happens when the ligand binds to the receptor, and it causes this conformational change. In terms of their structure, they have 7 transmembrane domains that span across the lipid bilayer, and they have an extracellular N-terminal domain, which is for where the ligand will bind. In this case, it's glucagon. And then they have an intracellular C-terminal domain on the other side of the receptor, if you like, that is bound to this heterotrimeric protein complex, and that heterotrimeric protein complex is made up of three constituents or three subunits: alpha, beta, and gamma. And it's the alpha subunit that is bound to guanosine diphosphate pre-ligand binding. And when ligand binding happens, that GDP will dissociate from the alpha subunit, and because guanosine triphosphate is more abundant in the cytosolic space, GTP will quickly bind to that alpha subunit, okay, replacing GDP essentially, and because GTP binding is favoured because of the conformational change that has happened because of, say, glucagon binding. Now, when that alpha subunit is bound to GTP, it becomes active and it actually dissociates from that heterotrimeric complex, leaving the beta and gamma subunits still attached to that C-terminal domain, okay, that intracellular C-terminal domain. That alpha subunit will move and it will bind to typically adenylyl cyclase, or adenylate cyclase as some people call it, and that's a membrane bound enzyme that's capable of generating the secondary messenger cyclic adenosine monophosphate from adenosine triphosphate. Now, and obviously CAMP can activate protein kinase A, and protein kinase A is really important in activating phosphorylase kinase, which is the regulatory enzyme that will activate glycogen phosphorylase, okay, and it can also inhibit glycogen synthase as well. Now, I mentioned before there's also GQ protein coupled receptors that glucagon's able to signal from, and on the other side of the diagram, on the left-hand side, you can see the alternate signal transduction pathway that can take place with GQ protein coupled glucagon receptors. So that, again, it's the same principle. That heterotrimeric subunit, that alpha subunit, will become activated upon ligand binding and guanosine exchange. So when GTP is bound to that alpha subunit, it will dissociate, and instead of activating adenylyl cyclase, it will activate phospholipase C. Now, phospholipase C is a lot of people might be familiar with that enzyme, but it's a really important enzyme in hydrolyzing your phosphatidyl inositol 4,5-bisphosphate, that phospholipid that's abundant in your cell membranes in close proximity to these receptors. Okay, so when you hydrolyze PIP2, you get inositol 1,4,5-triphosphate, which is also called IP3, and you also get diacylglycerol. Now, that inositol 1,4,5-triphosphate, or IP3, in biology it's renowned for being a potent calcium chaperone, so it facilitates calcium movement, if you like, and that calcium will bind to phosphorylase kinase. Phosphorylase kinase has a calmodulin regulatory domain, so when calcium binds, it becomes activated. Okay, so it allows for independent, it allows for the activation of phosphorylase kinase without having to activate protein kinase and CAMP2, and that secondary messenger CAMP. So it's really important having these two pathways. It allows for amplification, okay, and then you see the outcome is still the same though, because the outcome's still the same. Phosphorylase kinase will still become activated. It's either activated by calcium binding to its calmodulin regulatory domain, or it's activated by protein kinase A, and that protein kinase A is activated by your cyclic adenosine monophosphate, and of course protein kinase A will activate phosphorylase kinase too. And then once phosphorylase kinase is activated, it will then inhibit glycogen synthase via phosphorylation, and it will activate glycogen phosphorylase. And glycogen phosphorylase is the main enzyme that is responsible or facilitates glycogenolysis. Okay, glycogen's broken down by phosphorylase, okay, adding those inorganic phosphate groups to give you your glucose 1-phosphate. Now, the aim of glucagon receptor signaling is to increase hepatic glucose output, of course, because glycogenolysis, especially if we're looking at the liver, okay, the liver expresses enzymes like gluconeogenic enzymes like glucose 6-phosphatase, okay, and that's really important in increasing hepatic glucose output. Once that glycogen phosphorylase has broken down that glycogen into glucose 1-phosphate, phosphoglucomutase will transfer that phosphate group from the anomeric first carbon to that 6th carbon on the hexose ring, giving you glucose 6-phosphate, and then glucose 6-phosphatase can remove that phosphate group completely, giving you free glucose, and that glucose is able to diffuse out of the liver cells via the GLUT transporters into the bloodstream. Now, of course, hepatic glucose output isn't just achieved by glycogenolysis, it's also achieved by gluconeogenesis, and this is actually a really important concept to understand when when looking at blood glucose homeostasis, because of course, as I mentioned before, glucagon is secreted from those alpha pancreatic cells, not just in response to hypoglycemia, but also in response to high amino acids in the blood. Of course, many people like to be on, high protein, low carb diets. Bodybuilders tend to favor that. A lot of athletes like high protein. A lot of people who are trying to lose weight, there's the Atkins diet, paleo diets tend to be really high in proteins, but very, very low in carbs. So naturally, glucagon isn't just released in a fasted state. It's released when there are not a lot of carbohydrates available in the body, and your body can then start to convert those amino acids into glucose itself. As those glucogenic amino acids like alanine can be converted into glucose, okay, and that glucose can then be used to kind of raise blood sugar levels, or it can be used to be stored as glycogen again. And when you look at that, that's quite an important kind of, it's like a cycle if you think about it, and it's really important when you understand that when someone's on a high protein, low carb diet, they're not getting those carbohydrates, glycogen can still be stored in the liver, and glycogen can still be used to maintain glucose homeostasis in the absence of carbohydrates. So when glucagon is secreted and it affects the liver cell, it binds to these receptors in your hepatocytes. It will facilitate hepatic glucose output, not just by glycogenolysis, but also by gluconeogenesis. So glucagon will result in the activation of protein kinase A, and protein kinase A is really important in activating gluconeogenic genes like your PEPCK and your glucose 6-phosphatase. Okay. And of course, if gluconeogenesis is upregulated, then beta oxidation naturally is going to be upregulated as well, and lipogenesis will be inhibited. So again, it's that reverse of insulin signaling. And why is that? Because of course, gluconeogenesis is an anabolic process that requires a lot of ATP. But remember, we don't have high levels of glucose in the first place, especially if you're on a high protein diet or you're in a fasted state. But one way you can get that energy is through the breakdown of your fats, your stored fats. So beta oxidation is the breakdown of those fats to generate your ketone bodies like hydroxybutyrate. Okay, and that energy is used to facilitate gluconeogenesis. Okay, so it just kind of shows how good our cells are at adapting to what we're putting into our bodies, and how often we're eating, from an evolutionary perspective. And of course, I'm going to touch on all of this a lot more in the coming slides, but you can kind of see this diagram basically shows you, this is a really good diagram, it shows you the relationship the signal transduction pathways protein kinase A, glucagon, has with all of these different metabolic pathways. So you can kind of see like glycogenolysis, gluconeogenesis, glycogenesis, glycolysis, and of course you've got lipogenesis, you've got beta oxidation, and they're either the genes that either facilitate lipogenesis will of course be downregulated when glucagon is signaled, but then when insulin signaling happens, then lipogenic genes will be upregulated, and gluconeogenic genes and lipogenolytic genes will be downregulated. So you can kind of see there's a pattern here, and once you learn it, it becomes a lot more clear. So as I said before, the glucagon binds to that kind of N-terminal domain in the extracellular space, and you get that conformational change of the heterotrimeric stimulatory G protein, and that's got the alpha subunit as well as the beta and gamma subunits. But the alpha subunit is what will participate in the guanine nucleotide exchange. Okay, it's a GEF factor. Okay, so the GPCR is acting as a guanine nucleotide exchange factor because guanosine diphosphate on the alpha subunit of that kind of heterotrimeric complex is being replaced with GTP, and it literally just the GDP will dissociate because the conformational change that's caused by ligand binding, so glucagon binding that G protein couple receptor, will favor GTP binding over GDP, and of course GTP is abundant in these cells, so it will happen quite quickly. And then when GTP is bound to that alpha subunit, it's considered active, okay, and it dissociates from that heterotrimeric stimulatory G protein on the C-terminal in the kind of intracellular space, and it will bind to your adenylyl or adenylate cyclase, and that's a membrane-bound enzyme that's responsible for the catalytic formation of cyclic adenosine monophosphate. Okay, so it gets rid of that beta and gamma phosphate, and then it's able to kind of perform this kind of intramolecular cyclization. So you can see I've actually got a diagram because a lot of people get confused and they mix up, or they mix up adenosine monophosphate with cyclic adenosine monophosphate. And again, cyclic adenosine monophosphate and adenosine monophosphate, they both have that adenine nitrogenous base. They've got that ribose sugar and they have that alpha phosphate. The only difference in cyclic adenosine monophosphate, that alpha phosphate has formed a cyclic ring with that hydroxyl group on the ribose sugar, and you can see that there. You can see that oxygen on that, you know, phosphoric acid group. That hydroxyl group has kind of attacked that hydroxyl group on the ribonucleotide sugar, and it's formed this kind of like ring. Okay, and CAMP is a really potent secondary messenger. It's really important in activating our protein kinase A, and protein kinase A is really important in activating certain enzymes and transcriptional shifts that will facilitate glycogenolysis, gluconeogenesis, beta oxidation, and inhibit processes like glycogenesis, lipogenesis, and glycolysis. So before we kind of move on to glycogenolysis itself, I just want to touch on glycogenolytic signaling in muscle cells, because it is different to that in liver cells. And of course, in different tissues, glycogen serves different purposes. So in fat tissue, it's used as access to glycerol for triglycerides in those adipocytes, and in your hepatocytes, your liver cells, glycogen's used to maintain glucose homeostasis. So of course, glycogenolysis is initiated by glucagon signaling, and then when protein kinase A is activated and glycogen phosphorylase is activated, that glycogen can be broken down by phosphorylysis, and it will give you your glucose 1-phosphate. And of course, as I mentioned before, your liver tissue expresses glucose 6-phosphatase enzymes, which your muscle tissue does not. So glucose 6-phosphatase can essentially remove that phosphate group from that glucose 6-phosphate once phosphoglucomutase has converted that glucose 1-phosphate that was broken down in glycogenolysis into glucose 6-phosphate. Okay, and then that free glucose, that glucose 6-phosphatase, can remove that phosphate group, giving us free glucose to leave the cell, to leave the liver cells and into the bloodstream. So it increases that hepatic glucose output, and it will raise your blood sugar levels after you have hypoglycemia after fasting. Now, in muscle cells, glycogenolysis serves as a completely different function. It is quick access to ATP. So muscle contraction is a very ATP demanding process. So glycogenolysis in muscle serves as local ATP production during muscle contraction to kind of meet or facilitate energy demands. And of course, glycogen phosphorylase in muscle cells is not activated by glucagon, because muscle tissue does not express glucagon receptors. Instead, they express beta adrenergic receptors and alpha-1 adrenergic receptors. So glycogenolysis and glycogen phosphorylase is actually activated by epinephrine, or most people will know that hormone as adrenaline. And adrenaline will signal through two distinct G-coupled protein receptor pathways, but both will result in the activation of glycogen phosphorylase. And of course, glycogenolysis in muscle cells is also initiated by calcium influx as well. Muscle contraction requires calcium from the sarcoplasmic reticulum in those myocytes, and that calcium can bind to calmodulin, and that's actually a subunit of phosphorylase kinase, which is the enzyme that will activate glycogen phosphorylase. And the reason there is so much there's so many different signaling routes to glycogenolysis in muscle cells is because it is kind of all or nothing. When you think about it, you want absolute activation of the glycogen phosphorylase to get the most ATP out of that glucose, out of that glycogen. So if you have many different activation pathways, it essentially amplifies glycogen phosphorylase activation. And obviously, the more adrenaline, the more muscle contraction, the more glycogenolysis is going to be happening. And of course, if you're exercising a lot, if you're running long distances, or if you're lifting heavy weights, of course you're gonna release adrenaline, because you release adrenaline when you're exercising. You also release adrenaline in stressful situations from an evolutionary perspective. If you're faced in a survival situation, having that adrenaline and initiating that glycogenolysis and amplification of glycogen phosphorylase activation will allow for maximum ATP generation in muscle tissues. So glycogenolysis in muscle cells integrates several different signaling pathways in order to maximize the activation of phosphorylase kinase. and glycogen phosphorylase to maximize ATP generation to kind of facilitate energy demands during exercise, during stressful situations, you know, when you need to use your muscles. Now, epinephrine, as most people call it, adrenaline, can signal through multiple different receptor pathways, and in muscle cells it signals through the beta adrenergic receptors, the beta 2 to be precise, and the alpha 1 adrenergic receptors. Now, they're both G protein coupled receptors, but they actually initiate different signaling pathways. So the beta 2 adrenergic receptors is a GS coupled protein receptor. So upon ligand binding, that intracellular heterotrimeric subunit, that alpha constituent on that heterotrimeric subunit, is GDP bound, but when ligand binding happens, GTP binding is favored over GDP. You get a guanine exchange factor, okay, and guanosine diphosphate dissociates, and guanosine triphosphate will bind onto that alpha subunit. Of course, that alpha subunit becomes activated, and it will dissociate and bind to your adenylyl cyclase, or as some people call it, adenylate cyclase, and that's the membrane-bound enzyme in close proximity to these GS-coupled B2 adrenergic receptors. And that adenylyl cyclase is capable of producing cyclic adenosine monophosphate from adenosine triphosphate. It's a very clever enzyme, and that cyclic AMP will activate protein kinase A. Now, protein kinase A will phosphorylate phosphorylase kinase, and phosphorylase kinase is the enzyme that activates glycogen phosphorylase A. Of course, glycogen phosphorylase A is actually glycogen phosphorylase B when it's in its inactive form. So phosphorylase kinase, when it phosphorylates glycogen phosphorylase B, it will convert to its active form, glycogen phosphorylase A, and that enzyme is capable of glycogenolysis. It phosphorylates, or phosphorylysis if you like, the glycogen, adding inorganic phosphate, giving you glucose 1-phosphate. And then, of course, muscle cells, they don't possess the glucose 6-phosphatase, but they do possess phosphoglucomutase. So that glucose 1-phosphate in an ATP independent manner can be converted into glucose 6 phosphate, but via phosphate transfer. Of course, glucose 1-phosphate has that inorganic phosphate on the anomeric carbon on the hexose ring, whereas glucose 6-phosphate has that phosphate group on the 6th carbon of the hexose ring. So it's just a phosphate transfer, if you like, and it's ATP independent, and it gives you quick access to glucose 6-phosphate. As you know, glucose 6-phosphate is a starting point to glycolysis and ATP production. Now, phosphorylase kinase also inhibits glycogen synthase as well by phosphorylation, and that's really important to kind of understand that as well, to kind of prevent futile cycling, to prevent, obviously, the goal is to break glycogen down now, not to build glycogen up. So glycogen synthase is not needed, so to prevent it from going back on itself, it's also inhibited by phosphorylase kinase. Now, of course, as I said before, you have the alpha adrenergic receptors, which are GQ protein coupled receptors. Again, they still have that, structurally they have that 7 transmembrane domain, and they have that intracellular heterotrimeric protein complex with the alpha, beta and gamma subunits, and it is still the alpha subunits that is bound to GDP. And upon ligand binding, of course, epinephrine will bind to those alpha 1 adrenergic receptors, and again you still get the guanine or GTP exchange, the guanosine exchange factor. That GDP is exchanged for GTP. GTP binding is favoured on that alpha subunit. That alpha constituent will dissociate, but instead of activating adenylyl cyclase, it will activate the enzyme phospholipase C. Now, I mentioned before, these receptors are in the lipid bilayer, or the cell membranes of our muscle cells, and in muscle cells they also have high concentrations of your phosphatidylinositol 4,5-bisphosphate, and those phospholipids that are in close proximity to these GQ couple receptors, and phospholipase C is able to hydrolyze that PIP2 into an inositol 1,4,5-triphosphate and diacylglycerol. Now, of course, an inositol 1,4,5-triphosphate and diacylglycerol are both kind of potent secondary messages. IP3 in particular is a really potent calcium chaperone. So IP3 will bind to receptors on the sacroplasmic reticulum, which is an organelle in these myocytes, and when calcium is released, calcium can bind to that calmodulin on phosphorylase kinase, because calmodulin is a regulatory subunit on phosphorylase kinase. So when calcium binds, it activates phosphorylase kinase without needing protein kinase A, and that's the phospholipase C pathway. So so it's not necessarily dependent on protein kinase A activation and beta adrenergic G couple receptor signaling. It also goes through the A1 receptors and the hydrolysis of your PIP2 inositol to get your inositol 1,4,5 triphosphate. And IP3 in biology is one of the most potent calcium chaperones that cells can use to kind of chaperone and move calcium, if you like, and stimulate calcium migration. And of course, muscle contraction itself naturally causes calcium release. The mechanisms behind muscle contraction, I won't get into it too much, but it relies on that calcium influx and calcium release to initiate muscle contraction. So more contraction means more calcium release, and calcium will essentially activate phosphorylase kinase, because phosphorylase kinase has a calmodulin regulatory subunit to allow for calcium binding and activation of phosphorylase kinase. And of course, as we know, our phosphorylase kinase will inhibit our glycogen synthase to prevent futile cycling, but it will also activate our glycogen phosphorylase B to glycogen phosphorylase A and allow for glycogenolysis. So that's essentially glycogenolytic signaling in muscle cells. And I think the main takeaway is adrenaline will initiate glycogenolysis, and calcium-induced muscle contraction will also initiate glycogenolysis. And the reason we have so many different options in muscle cells is to kind of maximize signaling glycogenolytic pathways and activation of our phosphorylase kinase enzyme. And to maximize that activation, having many different methods to activate will essentially maximize ATP production in muscle cells to kind of facilitate those demands. So before, glycogenolysis is the breakdown of glycogen into glucose. It's glucose subunits, it's monomeric subunits. Now, in the liver, it mainly relies on protein kinase A activation, but of course, as we know, our IP3, our inositol 1,4,5 triphosphate, is a potent calcium chaperone, and that can also directly activate phosphorylase kinase, because phosphorylase kinase has that calmodulin regulatory unit. Protein kinase A can activate phosphorylase kinase as well, and obviously our phosphorylase kinase is able to activate our glycogen phosphorylase. So you have your glycogen phosphorylase B, which is the inactive form of the enzyme, but when it's phosphorylated by phosphorylase kinase, you get glycogen phosphorylase A, and that glycogen phosphorylase A is able to directly break down those alpha 1 to 4 glycosidic bonds by a phosphorylated addition of phosphorylysis, the addition of an inorganic phosphate group to produce glucose 1-phosphate, and then that glucose 1-phosphate can be converted into glucose 6-phosphate via phosphoglucomutase, and that glucose 6-phosphate can, in muscle cells, can be used for glycolysis. Okay, in adipose cells, in adipocytes, it can be used to generate glycerol. Again, that's still through glycolysis, which requires our glucose 6-phosphate as a starting point. But then, of course, in liver cells, liver cells also have those that shift in gluconeogenic gene expression, and one of those gluconeogenic genes is glucose 6-phosphatase. And glucose 6-phosphatase is really important in removing that inorganic phosphate group from that 6th carbon on the hexose ring to give free glucose, and that glucose is able to diffuse out of the hepatocytes passively through those GLUT transporters, okay, into the blood to raise blood sugar levels. Now, of course, this glucose 6-phosphatase is really important to remember that it only can remove those phosphate groups from the 6th carbon. So we need our phosphoglucomutase in all of our tissues, because that phosphoglucomutase is really important in both our muscle tissues, in our fat tissues, and in our hepatocytes or liver tissues to transfer that phosphate group from the anomeric 1st carbon to the 6th carbon on the hexose ring. So that phosphoglucomutase is really important. But of course, both fat cells and muscle cells lack that glucose 6-phosphatase, because the glucose in those cells isn't being used for output like it is in liver cells. And to get glucose out of tissue, out of cells, you need to remove those negatively charged phosphate group to allow passive diffusion out of the cells. Now, I just want to touch as well, as you know, glycogen has 1 to 6 alpha linkages as well, glycosidic linkages, and those linkages are dealt with by a debranching enzyme, okay, this transferase enzyme, and that enzyme has a hydrolysis domain, and it will hydrolyze those 1 to 6 residues, releasing free glucose. Okay, so glycogenolysis results in the generation of free glucose and also glucose 1 phosphate. So like with insulin, glucagon signaling will result in several transcriptional shifts that will essentially change cell processing to facilitate certain metabolic pathways and suppress other metabolic pathways. And when you understand how this works, it gives you a really clear picture on how our metabolism, especially in the liver, actually works, and how we look at glucose homeostasis, fat storage, what happens when we're fasting, what happens when we're eating lots of carbs or eating lots of protein. But glucagon itself will mainly signal through GS protein coupled receptors in the liver. So that leads to adenylate or adenylyl cyclase activation and the production of cyclic adenosine monophosphate. And that CAMP is really important in activating protein kinase A. Now, protein kinase A phosphorylates a certain transcription factor called the cyclic adenosine monophosphate response element binding protein. It's called CREB for short. Now, CREB combine to the CRE elements, okay, and that can recruit certain cofactors like your CBP. Now, this transcription factor will upregulate certain genes that are associated with gluconeogenesis, like your phosphonyl pyruvate carboxykinase and glucose 6-phosphatase. And this is really important because the role of glucagon signaling is to increase hepatic glucose output to raise blood sugar levels. And if someone is on a high protein diet, for example, even if they're not fasting and they're eating a lot of protein, those dietary carbs and sugars might not be available to draw from, but what we do have is a high level of amino acids in the blood, okay, and a lot of amino acid availability. So the carbon skeleton of those amino acids can be used to build sugars, build glucose. Now, phosphonyl pyruvate carboxykinase is a gluconeogenic enzyme that it's considered a rate-limiting enzyme. It's really important in gluconeogenesis. What it does is it converts the Krebs cycle intermediate oxaloacetate into phosphonyl pyruvate. Then, of course, phosphonyl pyruvate can be converted into pyruvate, and once you have pyruvate, you can essentially do the reverse glycolysis and build up that carbon skeleton to get your 6-carbon sugar. And of course, this is using the carbon skeleton from amino acids, particularly glucogenic or exclusively glucogenic amino acids like alanine. And alanine can be converted into Krebs cycle intermediates like oxaloacetate. And of course, that phosphonyl pyruvate carboxykinase is a really important enzyme in converting those Krebs cycle intermediates to those glycolytic intermediates, okay, and starting to actually build your glucose. And of course, hepatic glucose output can be achieved through gluconeogenesis as well as glycogenolysis, because if you think about it, once you build your glucose, once you get your glucose 6-phosphate from pyruvate, essentially the reverse of glycolysis, you have glucose 6-phosphatase enzyme which can remove that phosphate group from that 6th carbon and allow for glucose output out the cell. So it's through gluconeogenesis and through glycogenolysis. But of course, gluconeogenesis is an anabolic process, so it requires a lot of ATP. But people who are on high protein diets, or people who might be fasting or having low carbs, might not have that dietary glucose availability to generate ATP. So then, of course, beta oxidation will be increased or facilitated, so our fat stores can then be used to generate energy. So of course, glucagon will facilitate beta oxidation, okay, and the purpose of facilitating beta oxidation is to facilitate processes like gluconeogenesis when dietary glucose isn't as available. Now, beta oxidation is the breakdown of fats into ketones or ketone bodies like hydroxybutyrate to get energy. So your body goes into, say, ketosis, and glucagon can facilitate this. And one way it does this is by upregulating lipogenolytic or genes associated with beta oxidation and suppressing gene expression or lipogenic gene expression. Now, if you remember, insulin signaling resulted in the phosphorylation of your FOXO1 transcription factor. Now, in glucagon signaling, FOXO1 remains unphosphorylated, it remains activated. So hyperglycemic gene expression is suppressed. So genes like acetyl CoA carboxylase is suppressed, and of course that means that malonyl CoA production will be essentially decreased, and malonyl CoA inhibits beta oxidation and inhibits long chain fatty acid transport into the mitochondria. So when malonyl CoA concentration is down, beta oxidation can, those long chain fatty acids can get into the mitochondria where they can be broken down. But also lipogenic genes as well will be suppressed, because the aim here is to break down fats, not build them up. And it's again like glycogenic genes will also be suppressed, because the aim is to break down glycogen, okay, not build glycogen from glucose. So this kind of prevents futile cycling, and you can kind of see the relationship these metabolic pathways have with each other. That beta oxidation, that hydroxybutyrate, those ketone bodies you generate from beta oxidation are used as energy to facilitate processes like gluconeogenesis, okay? And the amino acids, from dietary amino acids or glucogenic amino acids like alanine, their carbon skeleton can be used to be converted into Krebs cycle intermediates. And then when you get Krebs cycle intermediates like oxaloacetate, you get rate limiting enzymes like your phosphonyl pyruvate carboxykinase to kind of convert that oxaloacetate into phosphonyl pyruvate, and then you have your glycolytic intermediates, and then you can build your 6-carbon sugar. And of course these changes in transcriptional shifts, these changes in gene expression and enzyme expression, facilitating, say, lipogenolysis or beta oxidation, gluconeogenesis, glycogenolysis, and then suppressing lipogenesis, suppressing glycogenesis, kind of makes sense when you're looking at what the goal of the body or what the goal of the cell is. It kind of makes sense. So I've got this table here just to kind of compare and contrast the effects of insulin and glucagon and the functions they have, where they're secreted and synthesized. And it's a good table to kind of copy down, because it's, everything we've spoken about is summarized here onto this table. So like, for instance, insulin is secreted by beta pancreatic cells in response to hyperglycaemia, and glucagon is secreted by alpha pancreatic cells in response to hypoglycaemia. Both beta and alpha pancreatic cells make up the islets of Langerhans in the pancreas, along with their delta cells. Now, insulin facilitates glucose storage in muscle cells, fat tissue, and liver tissue. Okay, so hepatic glucose output is decreased and hepatic glucose input is increased. So as you know, when insulin binds to those RTK receptors in the liver, there's going to be an upregulation in GLUT4 expression and translocation to the membrane to allow for more glucose to go into those cells. Okay. Glycogenolysis, however, is mainly facilitating hepatic glucose output in liver cells and is used in muscles and fat tissue for slightly different reasons. So in fat tissue, glycogenolysis is used to access quickly glucose 6-phosphate to generate glycerol for triglycerides. In muscle cells, it's used to access glucose 6-phosphate quickly for glycolysis and cellular respiration to generate a lot of ATP to meet ATP demands, especially during exercise, and or stress, fight or flight situations. Now, both insulin and glucagon signal through 2 different receptor families. Insulin depends on the receptor tyrosine kinase family, okay, and glucagon depends on the GQ and GS metabotropic protein or G protein receptor family. So insulin results in the activation of protein kinase B, okay, whereas glucagon results in the activation of protein kinase A, okay. So what's being activated, the regulatory enzymes, the regulatory kinases are very different. One's protein kinase A in glucagon, and insulin is protein kinase B. Now, insulin results in the upregulation of glycolysis. It results in the upregulation of lipogenesis. Okay, and it results in the suppression of gluconeogenesis, the suppression of beta oxidation, and the suppression of glycogenolysis. Okay, whereas glucagon results in the upregulation of beta oxidation, the upregulation of gluconeogenesis, the downregulation of glycolysis, the downregulation of glycogenesis, and the downregulation of lipogenesis. Okay, and how does this happen? Okay, what's happening? There's transcriptional shifts in gene expression that suppress certain rate limiting enzymes. That's the key. So certain transcription factors like FOXO1 in insulin signaling becomes phosphorylated, whereas in glucagon signaling it does not become phosphorylated. Okay, certain gluconeogenic genes are upregulated during their expression is upregulated during glucagon signaling, such as your phosphoryl pyruvate carboxykinase, okay, and your glucose 6-phosphatase. Whereas in insulin signaling, genes like glucose 6-phosphatase and your phosphoryl pyruvate carboxykinase, their expression is downregulated or suppressed. Okay. And it's the same with, lipogenic genes. For example, your acetyl-CoA carboxylase is really important in insulin signaling because it increases malonyl-CoA production. Okay, acetyl-CoA, that acetyl-CoA carboxylase is able to convert acetyl-CoA into malonyl-CoA. Now, malonyl-CoA inhibits fatty acid transport into the mitochondria, so it will prevent beta oxidation. But malonyl-CoA is a rate-limiting step in initiating fatty acid synthesis, so lipogenesis. So you can see, and I'm going to talk about this more in the coming slides, but you can kind of see the differences. The insulin and glucagon are essentially the antithesis of each other. So insulin stimulates glycolysis, it upregulates glycolysis, and it makes sense because the role is to facilitate glucose storage, but of course we can only store so much glycogen, we can only store so much fat, and we need a lot of energy to do glycogenesis, and we need energy to do lipogenesis. So that energy comes from glycolysis. So insulin stimulates glycolysis by upregulating the expression of glycolytic enzymes like glucokinase, and a lot of people know glucokinase is really important in phosphorylating that glucose to glucose 6-phosphate. So when hepatic glucose input is increased, you get more free glucose moving into these hepatocytes. It can be glucokinase activity is upregulated, so you're going to get more glucose 6-phosphate production. Okay, and glucose 6-phosphate production is really important in both glycogenesis and also glycolysis. Okay, and another enzyme is phosphofructokinase. That's also upregulated, and that's a rate-limiting step in glycolysis where fructose 6-phosphate is converted to fructose 1,6-bisphosphate, and then that fructose 1,6-bisphosphate can then be split into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and that's a really important step in glycolysis. So that enzyme is also upregulated. Pyruvate kinase is also upregulated as well, so you get more production of pyruvate. Now, pyruvate can then be used for lipogenesis, or it can be used for cellular respiration as well to get more energy. So glucagon will actually inhibit glycolysis, and it makes sense because we're trying to facilitate hepatic glucose output, so we don't want that glucose being broken down. So it does this by inhibiting the transcription of certain glycolytic genes like glucose kinase, but it also does this by protein kinase A. As you know, glucagon will activate protein kinase A. Now, protein kinase A can phosphorylate a bifunctional enzyme called phosphofructokinase 2/fructose 2,6-bisphosphatase, and depending on what domain is inhibited will directly depend on fructose 2,6-bisphosphate levels. Now, this enzyme is considered a master regulator of both glycolysis and gluconeogenesis. So when fructose 2,6-bisphosphate levels decrease, it will reduce the activity of your PFK1 enzyme. So that was the end of part 1. In part 2, we're going to go over neural control. We're going to go over the more transcriptional changes induced by insulin and glucagon and how that affects pathways like beta oxidation, gluconeogenesis, and then in part 3 we're going to look at glycolysis and gluconeogenesis. I hope everyone found this video helpful.